ready?
Yes.
No, and sometimes we feel like we should because then we don't have to worry about. I think we have a headset, but those are a little bit annoying to wear. I don't know if you
Yeah, they are.
Can you hear this? Sounds a little loud. We'd like to welcome our streaming audience.
Live from the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh, it's the Science Cafe with your host, Brian Mallow.
Good evening, everybody. Hello. Welcome to the Daily Planet Cafe here in the Nature Research Center of the North Carolina Museum of Natural Sciences. So it's Thursday night, which means it's Science Thursday, and it's our Science Cafe. Is anyone here for the first time for our cafe? Welcome. Thanks for coming out. What brought you out here? Was it the topic or the speaker, or you knew that guy? He's the guy. Excellent. Why are you here? You're interested in the research. Excellent. So, we do this every Thursday. The first Thursday of each month, we have science trivia night. It's games. People compete in teams, and it's a lot of fun. But all the other Thursdays are science cafes and quite a variety of topics, which I forgot to check the list. Katie, what topics do we have coming up? We still have sharks coming up, rabies. Next week is science trivia night, right? It's trivia night next week, and then rabies and sharks and quite a variety. So we try to cover all the sciences. We recently had really great programs on Pluto and the New Horizons spacecraft, and another one on the 25th anniversary of the Hubble Space Telescope. So we try to cover everything, and it's usually a lot of fun. It's basically a short presentation. Not quite a lecture. We're in a cafe. Food and beverages are available throughout the program. Restrooms are right back here. It'll be a relatively short presentation, and half our evening is Q&A. So it'll be about an hour program, and we count on you and your interest in the subject. We expect some very interesting questions from you. All eyes are upon you. All right, maybe that's an exaggeration. But so we have been looking forward to this program for a long time. It's an interesting. I wasn't aware that this much and this level of research was going on at UNC. So UNC is a center where they're working hard to come up with cures for HIV. Our guest tonight is a professor of medicine and immunology and epidemiology, and he is the director of the UNC HIV Cure Center. How about a nice round of applause for Dr. David Margolis?
Thanks very much for having me. HIV is actually a new disease. Historically speaking, it's only been in the human species for maybe 6 or 7 decades. And it's actually a very good example of how science has transformed medicine. This is a disease where when it first broke across the world in the 80s, if you were 25 years old and you were infected, there was a 50% chance you'd be dead in 2 to 4 years. And then over about 15 years, antiretroviral therapy, antiviral medicines that stop HIV disease were developed. And now the life expectancy, which is on this graph, of a person with HIV infection is essentially not any different from someone without it. And so this has led to a lot of new recommendations that came out from the Centers of Disease Control, and in fact the White House just this week, that we need to do a better job of finding people with HIV and getting them on treatment to prevent disease in them and spread of HIV infection, both here and everywhere across the world. And so this is the AIDS quilt in 1996, and it's now too big to be put down in any one place since 1996. But we really are in a paradigm of trying to find people with infection and treat them and prevent new infections. But there are hundreds of millions of people in the world with HIV infection, and so one of the things that's emerged in work recently is the idea that maybe we can figure out a way to cure infection, to eliminate infection in a person, and add that to our tools to shrink the effect of the pandemic across the world and maybe someday get rid of HIV altogether. But that's a very difficult problem. And so the first step is really thought to be the prevention and treatment step, and that's what you read about in the newspaper. I think the statements from the White House Office of AIDS came out just today, talking about finding people with infection and preventing infection by treating them. This was actually a study led by another person at UNC, Dr. Myron Cohen, a big paper in the New England Journal of Medicine called the Science Breakthrough of the Year in 2011. And the idea is the more people we can get on treatment, the less chance there is for transmission. And now it's been shown that treatment of a population in an area actually reduces the incidence of AIDS transmission over time there. But that leaves us with a lot of people to treat. And since we can't cure infection yet, people would be treated forever. And so part of the reason that the idea about developing new therapies to cure AIDS came in. Another thing happened, and this was a newspaper article in 2008, where a man in Germany who had HIV but also had cancer got a bone marrow transplant to treat his cancer, and unexpectedly, the bone marrow transplant cured him of HIV. He is the only person in the world who's ever been cured of HIV, and the reason is the cells that he got from his transplant were genetically resistant to HIV. And so this was something that wasn't thought to be possible. And so these sort of two events, the social event, the medical need, and the event that, and this idea that it's possible, has really galvanized the field over the last, I would say, 8 or 9 years to really start working on this problem seriously. But we can't give millions of people bone marrow transplants, obviously, and so we need to figure out practical, safe, and affordable ways to eradicate HIV infection. So why doesn't therapy cure AIDS? If you have a bacterial infection, a pneumonia, and we give you antibiotics, it kills the bacteria and you're all better. HIV is a virus, and unlike many viruses, all viruses are parasites and live within, use cells to live, but HIV actually stitches itself into the genetic material of the human cell. It's what's so-called a retrovirus. And because of that, it becomes essentially like a cancer gene inside the cell and will stay there as long as the cell is alive. And so there are a few cells in the body called memory T cells. Their job is to live a long time. They're your immunological memory. That's why you remember seeing tetanus or bacteria or some pathogen and you have immunity. If HIV, which it does rarely, get into these cells, it stays there as long as immunological memory is around. And so this is a study measuring the frequency of that virus in those memory cells in a group of people over time. And you can see that pretty much the virus doesn't go away. And so the estimate from this study was that it would require more than 70 years of therapy continuously to cure someone just with therapy. So that's obviously not practical. How does this happen? Well, these are different kinds of T cells. A naive T cell hasn't seen anything yet and has to learn what its immune response should be. But when it sees an antigen and becomes activated, it then expands and does its job to clear out things that don't belong. These are the kind of cells that HIV is designed to infect. And so most of the time when the virus goes into these cells, it kills the cell or the cells die of other natural causes. But extremely rarely, when that cell becomes a memory cell, which is its job to become a memory cell to give you immunological memory, HIV is trapped along for the ride. And so this is where we think most of persistent infection resides. And this is what we think our target has to be in order to cure infection over time. And so this is our current idea of how things work. This is essentially a picture of an HIV positive person who's healthy, has a normal immune system, and they've been on therapy, and their therapy has blocked the spread of virus in their T cells. So they have what's called an undetectable viral load, but if you get cells from those people, you can recover virus from them. And if you stop their therapy, the virus wakes up, spreads, and disease begins again. So we think that there are, you can see on the right-hand side of the slide, those are the latently infected T cells where the virus is sleeping. It can't be treated by drugs because it's not doing anything, and it can't be cleared by the immune system because it's not making any viruses that can be seen. So we think we have and you can see on the left-hand side, although there may be a few viral particles floating around at this point, the drugs appear to be completely protecting new unhealthy cells from being infected by these few viral particles. So the disease is essentially stopped by therapy but not cured. So we think we need a two-step approach to cure infection. And the first step is to develop what we are calling anti-latency therapy. It's really cell-directed therapy to somehow signal the cell to force the virus out of hiding, to make the virus visible to the immune system, and then we would give a specific immunotherapy of some kind, and there are many examples of this, that would help the immune system clear the infected cells. And then there would hopefully at the end be no virus left. We could stop antiretroviral therapy and we'd be done. So how do you turn a sleeping virus on? It turns out that a sleeping virus is sort of actively sleeping. The virus is programmed to grow and replicate, but there are things in the cell that make it hard for the virus to grow all the time. And these are different molecular obstacles. We think that our therapies should target these obstacles, take them down, and allow the virus to express itself as it naturally does. And if we can do this in a careful way without messing up the cell machinery too much, we think that we will unveil the sleeping virus and then it will be able to be targeted and cleared. So this is a picture of a cell, and it illustrates what we were trying to do in an actual experiment that we were actually able to do in patients a few years ago. So you see, this is a memory T cell. It's in the resting state. It has the HIV genome inside its chromosomes, but there's nothing coming out of the virus. The virus is asleep. And what we would want to do is we would want to detect the signal of the latent virus leaving latency and making viral RNA message, making viral gene expression, which would then lead to viral protein expression and viral production. And so we took patients on therapy, sampled a lot of their blood cells, and measured the amount of HIV RNA gene expression in this resting cell population. And you can see this is not really a good slide to show you because all the letters are too small, sorry. But in the yellow bars is the level of virus expression that we could measure in pools of cells at one moment in time. And then we gave each of these 8 patients a single dose of a drug that slightly alters gene expression and seems to have a specifically broad effect on HIV. And you can see in all of these 8 patients, there's evidence of the virus waking up. So we don't know if this is enough waking up to allow the virus to be cleared, but it shows that we can do this with a medicine in the clinic in a safe way, and we can start to go down that path. So I talked about reversing latency and inducing virus expression. And the second step, we think, needs to be some sort of immune therapy to help clear infection. And I'm sorry, the room is too big and my slides are too small. I'll know better next time. But these are all different examples of ways that we already know how to help the immune system see and clear HIV. These approaches, which I won't go into detail, all come from vaccine research. So there's been attempts to develop HIV vaccines for more than 20 years now, and they have various different approaches. All of them seem to be effective to some extent, although we haven't yet invented a vaccine that's completely protective against HIV infection. But we think that our task is a little easier than a preventive vaccine because we only have a very few cells to clear. And the virus can't get away because patients are on therapy and the virus can't continue to grow and mutate and escape the immune response. So we think that a step-by-step approach of developing therapies that can expose the virus and then using the many tools that we have to augment the HIV immune response may help us clear infection. So I told you about an experiment in man that showed something, but we can't do risky experiments in people, particularly as therapy is quite good, they're very healthy, there's really no reason for them to risk anything as drastic as a bone marrow transplant. So we have to develop model systems. There's a virus like HIV called simian immunodeficiency virus, so we can study that in monkeys. And that's good, although it is a different animal, a different immune system, and a different virus. And now, with my colleague Victor Garcia, also at UNC, we have something called the humanized mouse, where an immunodeficient mouse who's lacking parts of its immune system actually gets transplanted with human cells that reconstruct a human immune system inside the mouse. And so here in the graph, you can see the mouse is exposed to HIV, virus spreads in the mouse, and the viral load goes up, and that's the graph there. Then we put the mice on the same therapy that people use, and you can see in the orange bars how the therapy works. Viral load drops. And if you stop therapy in the bottom corner, you can see viral loads jump up again. So now we can study combinations of drugs that we couldn't give to people at this point in the mouse and study all the human cells that we recover from the mouse and see how much HIV is left and how the drugs and approaches worked. Eventually, we'd like to get to this sort of a point with people once we have assays that work that can measure these very rare events and effective interventions. Take people on therapy, give them some drugs, give them maybe a vaccine in combination, measure what happens, and see how much progress towards eradication we've gotten. So you can see this will probably be a step-by-step process that will probably take many years. People always raise their hand and ask me, how many years? And I always say, more than 5. But probably longer than that. We really don't know. I can only say that we've only been working on this a short time. It took almost 20 years to develop effective therapy, and I wouldn't be surprised if it takes at least 20 years to develop effective curative therapy. So I think we are beginning to design and discover and develop the tools that will allow us to make gradual progress towards this goal. It will be probably very complex because most importantly, the most important message is that therapy for HIV infection is very good, and every single person in the world who is exposed to HIV, who has HIV infection, should be treated, and we should find ways to diagnose them and get them into medical care and restore their health. And that will be good for them and good for society. But ultimately, we should be able to figure out ways to unmask the virus and then clear it with the help of the immune system. So with that, I will just sort of wrap up and ask for any questions. Thanks.
Thank you. How about a nice round of applause for David Margolis? And yeah, we can just leave that up there. And yeah, so the rest of our program will be Q&A, so we count on you. Katie and I, we got some water for you, David. And Katie and I have microphones. Because we stream our programs, they go live on livestream.com, and then they'll be online later. We like to have all our questions on mic. So Katie and I have microphones, and if you raise your hands, we will try to get to you in basically the order you raise them. So we have a question in the back.
Good evening. It may be semantic, but what is the difference between AIDS and HIV?
It's sort of semantic, but it's sort of important. So AIDS was a term that was historically coined before we knew what HIV was. So AIDS is a medical term, acquired immunodeficiency syndrome. HIV is the virus that causes that disease. People get really wound up about whether I have AIDS or not, or did I have AIDS, or could I get AIDS? And that's really, I think, a lot around the stigma of the condition. Basically, if people are able to access medical care and get medication, there's really no such thing as AIDS. It's HIV infection, and if we can give you treatment, your immune system should get better, things should be fine. So on sort of the social medical context, it's a really big problem and burden for HIV-infected people to be get medical care and be treated. And there are patients that come to me to see me in the clinic in Chapel Hill from 2 hours away because they don't want anybody in their town to know that they need to, be on this treatment, that sort of a thing. So I think it's just very important to know that it's just a virus.
David, since related to that question, there's always been this fringe group of people that have said that HIV doesn't cause AIDS. Is there something you could tell us about that? Why would they think that? And can you address that in any way?
It's unfortunate. There's in a lot of areas there are these ideas that I guess at a point in time there was some rational reason to have a scientific question that maybe we don't really understand everything, but this is probably one of the most studied and understood viruses in the history of man. I really don't hear that many people that have any credibility raising those questions anymore.
I've heard a lot about immunotherapy recently, and doctors have talked about training T cells. And I have a real hard time grasping the concept of training a T cell. Could you comment?
I can. I'm more a virologist than an immunologist, but I'll try. We're actually doing a study where we're training T cells. We take blood from patients. We send them off to a special lab that trains the T cells, and we've just started giving them back to the T cells. So T cells learn to see an antigen, that's a shape, by exposure to it in the context of being taught to see the shape by other cells called dendritic cells and being helped to become specially responsive to that antigen, that shape, by the presence of cytokines, which are immune hormones. These are all things that naturally go on in our body, but now that we understand them, we can mimic them in the lab. So that's one way to train a T cell. The other way is just the usual vaccine. So if you put an antigen into a person, if it's a foreign antigen and if they're able to make a response to it, they will. So you can take pieces of HIV protein and mix them up and put them in a vaccine, and you'll get responses against these proteins and shapes and antigens. The problem with making an HIV vaccine that prevents infection is that there's so many shapes that HIV displays and it changes all the time. It's very difficult to make a vaccine that blocks every virus that could possibly be there. But the virus that a person has, we may be able to identify specific parts that are very conserved. That is, they're the same between all sorts of different viruses, and we may be able to target them specifically. So there's a whole lot of ways to train the immune system, not just T cells, and we're really exploring as many of them as we can.
excuse me, I guess a two-part question. First, this therapy would be sort of the second stage for a person after their virus was controlled, and then they would go on to sort of have this curative therapy as the goal.
Who knows, but that's the way we're thinking about it now, yes.
So then my question would be, and you go to look at this on a worldwide scale, what percentage of the world is sort of already at that stage where their virus is controlled and could go on to that, and what percentage is still uncontrolled?
So I think you're alluding to the fact that it's a very big problem to treat the many millions of people that need to be treated, and I agree with that. I think we've made a lot of progress, but there's still a long way to go. Treatment is getting much easier. There are now many kinds of treatment regimens that are made in a single combination pill that you take once a day, and that's all you need to take. There are newer treatments coming out that will probably be available in the next few years that are injectable, long-acting treatments, and you could also choose to get one shot every month or two. But you still have to deliver that therapy. You still have to monitor that therapy. That's something the medical delivery system has to do. I would just remind people that 10 or 15 years ago, nobody wanted to treat patients in Africa because it was just impossible. It was too hard. Now there are millions of people being treated in Africa, and the incidence of AIDS, of HIV infection in Africa, has actually gone down for the last 2 years. So you're right, it's difficult, but unless we have these aspirations, we'll never get there.
As we saw on the slide with the mice that had gone on therapy, and then we saw as they came off, the rate of infected cells rose. Is it possible that with taking people off of current therapies and the reactivating the latent cells, that there would be an increase in productivity of those cells then as they're reawakened? Or is it thought that it would be the same rate of multiplication at that point?
So we generally think that if treatment is stopped in an appropriate way, that when it's restarted, it generally seems to work just as well. So in the studies that we do, we're very careful about that. And we're very cautious about interrupting therapy because you do have a concern that somebody's on a regimen that's working, and you do something to them experimentally and you stop it and you try to restart it, it doesn't work as well, they've lost, that's a bad thing. But I think so far all of the evidence says that if this is done in a monitored way, in a controlled setting, generally, you don't develop drug resistance just by having the viral load come back.
All right, so 2 questions. So you said that the person had the bone marrow transplant had been cured of HIV. Has this been attempted again, and would it matter whether you replaced a different body part, like an arm or something? Would that matter?
So yes and yes. So this has been attempted several other times, and in fact, it's an ongoing study in patients that happen to have cancer needing a bone marrow transplant that have HIV. Some of them are being asked if they want to participate in another experiment like this. There were at least 3 or 4 patients that I know of where the transplant process did appear to greatly reduce the amount of virus inside them, but disease came back. They weren't cured. So it's not really clear exactly how it happened in Timothy Brown, who's the man who was cured. We think it's a combination of effects, and it may be something he was just sort of lucky that it only happens 1 out of 10 times, and he happened to be the lucky one. Your second question is, what's the transplant have to be? Well, mostly we think that we know that HIV infects a certain kind of white blood cell called the CD4 T cell. So we think that those cells are the ones that have to be transplanted because they have to be genetically protected or protected from infection in some way.
And then I've got one more. Sorry. Why was the person? Why was he like immune / repellent to HIV?
So the transplanted cells weren't Timothy Brown cells; they were someone else's cells. When you get a bone marrow transplant, you get cells from another person, but they look at the genetic makeup of you and the other person and make sure they're a reasonable match, just like matching a kidney or a heart or a liver. Bone marrow needs to be matched too. By chance, there's a genetic mutation that was probably happened in the Middle Ages or even earlier in Northern Europe. So 1 in 100 white Northern Europeans have this genetic mutation in both of the alleles of their genes. And if they have both mutations, the receptor that allows the virus into the cell, that the virus uses to get into the cell, its shape is changed and the virus can't get in. So their white blood cells are resistant to HIV. So there are, if you go to Germany, probably 1 in 100 people on the street can't be infected by HIV, just naturally. So it just so happened that Timothy Brown was matched exactly to one of these people, could accept a bone marrow transplant of their cells, and then those cells became resistant to HIV. It was very surprising to the field because we thought, and some people still think, that virus also hides out in other places besides just the CD4 T cell. But it does appear that he was cured, and there are people that are actually trying to do research to figure out how to engineer anyone's cell, genetic engineering for anybody's cells, to be HIV resistant, and could we sort of do that on a larger scale? That's really much more a technical challenge than a scientific challenge.
I have 2 questions that maybe you've answered. The first is, what is ART? And the second is, once you tease out the virus that's latent, how do you kill it?
Well, the first question is very, you must be a lawyer or something. ART is antiretroviral therapy. And how do you kill the virus once you tease it out? Well, there's a variety of ways. You could do it in a natural way or a synthetic way. So the natural way that cells are killed are usually by the white blood cells in the body that recognize foreign substances and either and release various chemicals that they naturally make onto those cells. Those chemicals poke holes in the cells or kill them in other ways. But we also have synthetic ways to kill cells. You could make, for instance, an antibody that is chemically linked with a toxin. And if that antibody sees an HIV protein and binds to the cell that's expressing HIV protein, it also brings the toxin into the cell and kills it. And that's actually been done in mice, but not in people yet.
All right. We know that viruses mutate a lot.
Some viruses mutate a lot.
The people that do vaccines have always expressed, and I'm talking about HIV, the people that do vaccine research seem to worry about that. Do you guys worry about that?
We worry about that to some extent. I think we have to worry about it less. And this gets back to this other young lady's question about people being on therapy. I think being on therapy as the first step towards cure is very important because it reduces the number of infected cells and it basically holds the virus in place. If the virus isn't growing, it can't mutate. So the sooner we get, and we actually have studied some patients that were put on therapy within days of infection, and we've measured by the genetic sequence of their virus, they have very little mutations, few mutations, because their virus hasn't been growing long enough to go very far. So And this is a, sort of a broad principle. But there are also ways to broadly target viruses despite their mutations. I talked about something called conserved epitopes. A protein can mutate, but at some point when it mutates, it doesn't work anymore. So there are parts of the virus that can't afford mutations because if they get mutated, the virus doesn't work. If we can target those parts of the virus, which for various reasons is difficult, then it wouldn't really matter about other mutations in other places. We'd still be able to see the virus.
Okay, there's two questions I have. One, do they know what, and I know that there's been some babies that were born to mothers who were HIV, okay, positive. The babies were shown at birth, at the moment of birth, to be HIV positive, but 2 or 3 days later were shown to be completely free of the virus. Do they know, and I know that these kids are being monitored and being tracked to see what it is they got. Obviously, they have something that allowed their system to kill off the virus and keep it from overtaking.
So
do we know anything? What is that? Or have we come any closer to understanding that?
So there are several cases like this. It sounds like you're talking about a case called the Mississippi baby. Is that what you're thinking of?
Yeah, there's
So the Mississippi baby was a very instructive, although in the end, sort of unfortunate case. So this was a child whose mother didn't have any prenatal care. She was HIV positive. She wasn't treated. She came into the hospital. They discovered she was HIV positive while she was in childbirth, and they started her on therapy at the time, and they started the baby on triple therapy within about 4 hours of birth, which is unusual because it's usually thought that that sort of treatment is too toxic for an infant. Nevertheless, the child was shown definitely to be HIV infected, and so there was virus recovered from the baby within the first few days of life, and the child was put on therapy and sent home, thought to be an HIV positive baby. For other reasons, the child was treated for about 16 months, and then the mother stopped therapy. There were social problems going on. When the baby came back to medical care, there was no trace of virus. And the baby was followed for 26 months. And for 26 months, I actually thought that the baby had been cured. Maybe treatment had been so early that latency hadn't been established. And once you just blocked the actively infected cells, there wasn't anything left to do. It turned out that 26 months later, the virus came back. So it may have been only 1 or 10 cells that remained in the child, but virus rebounded. So that taught us, I think, that this latent reservoir, even though it's very rare, we'll have to go find it and get it.
My second question is also, is there any way that they could take some of these group of cells and take them and sort of try to kill off the virus and make it like a serum that they could inject that would take care of all the virus in the person's body?
We're trying to figure out stuff like that, and I think there's a Dr. McCoy on a starship that can do things like that.
Yeah, I mean, I was just
we're
curious
not quite
because
there yet.
there's something not to explain, if you will, in greater detail, the guy that was cured in Germany. Explain exactly how was he cured of it?
Well, I think I said earlier, we think that it had a lot to do with giving these HIV-resistant cells, but he also got chemotherapy, radiation therapy. He actually had 2, not 1, bone marrow transplants because his cancer came back. So it's a very complicated case, and we don't exactly understand it. There are at least 2 other patients that were transplanted in Boston, and they had almost everything that Timothy Brown had done, but they were not cured. So we don't exactly understand it yet.
So I have 2 questions, and sorry if they get a little technical, but is the point of integration predictable where HIV
Yes and no.
itself?
So the virus has, if you want to know exactly, 52 sites in the human genome where it prefers to integrate. And there's a sequence preference for integration, but not a specificity. So the virus can be found almost anywhere, but is usually found in open areas of active genes. So it probably has to do with, as the virus is entering the nucleus, it finds an open spot and lands there, and some genes tend to be more open than others, and that's probably why there's the preference.
And then my other question is, it looked like you were using HDAC inhibitors
Yes.
to turn on genes. If you do that, are there other consequences because those are pretty unspecific that you're turning on a bunch of other suites of genes?
So we are using histone deacetylase inhibitors. These are drugs that inhibit an enzyme that controls how open or closed genes are and whether they're easier or harder to turn on and off. And so, yes, this is a host-directed therapy, and so there's some potential risks of that. So far, our studies don't really show any measurable toxicity of the HDAC inhibitors at the dose and the way we use them. There certainly are long-term possible issues. And I guess this is a sort of a risk and benefit. If it works and you only have to be on HDAC inhibitors for 6 months, maybe it's better off than having HIV infection and being on antiretroviral therapy for 60 years. We'll just have to see. But this is really our first step, and there are other general approaches of the same general nature, and a very big issue is the exact one that you bring up. Is this safe?
Can you comment a little bit on the technical reasons behind the difficulty in targeting the conserved epitopes of the HIV virus for vaccine development?
There's a lot. So many of these epitopes are so-called glycosylated. So a protein can have sugars laid on the outside of it, and HIV seems to have evolved to cover a lot of its important weak spots with these sugars. So that's one reason. And there are others, but I'm probably not a good enough immunologist to explain it.
Hey, David, can I ask you why are you in this field? So you're a virologist. How did you end up in HIV, and what keeps you in it?
Yeah, so I'm an infectious disease doctor. When I left college, there was a newspaper article. When I left college and started medical school, there were newspaper articles about gay men in Los Angeles dying of these weird infections. And so the HIV pandemic essentially sort of overlapped my entire medical career. And I always was interested in science and how science can be brought into medicine to make medicine better. If HIV is a really amazing example of this. If HIV had gotten into the human population 20 years ago, 20 years before it did, we wouldn't know the cell that it infects. We wouldn't have even defined it. We wouldn't be able to measure the virus in blood, and we wouldn't understand anything about it. So everything we understand about HIV is really from science that has developed within the last 25 years.
Sorry, I'll bring this over here. Beth, you have a question?
Okay, I have two questions. One, would you give us just a quick primer on why HIV has challenges with developing a vaccine? I've heard little bits about that, and I've read little bits about that. And then the second part is you described this unmasking of the cells hiding out. Is that something that would have to be repeated, or can you basically feel comfortable that you've unmasked everything at once?
And David, can I ask, just as far as that first part about what's challenging about coming up with a vaccine for HIV?
You should have Dr. Haynes from Duke come.
Why? What would he bring to it?
He's leading a major effort centered out of Duke to develop an HIV vaccine. So
what I wondered there was just like Beth, but my thought was some viruses are easy to kill.
Yes.
So why are some viruses easy to kill, and why is HIV not easy to kill?
Well, that's there's probably a lot of different answers to that, but the easiest one to say is that this is what's called this is a cross-species event. This is a monkey virus that has come into the human species. And this happens across all of nature all the time. When the West Nile virus got in that airplane in Egypt and landed in JFK about many years ago, 10 years ago or so, all of the birds along the East Coast got bad West Nile virus and died. And then they developed immunity to the new bird virus that now can sometimes get into people. HIV is a monkey virus that's evolved in the monkeys for millions of years. So most HIV monkey viruses are specific for a species of monkey. Those monkeys generally don't get sick from that virus. The monkeys get sick when they get a different chimpanzee's virus or a different macaque's virus. And humans get sick when we got exposed to these simian immunodeficiency viruses, which essentially adapted to grow into people. So that's one reason. There's a lot of more complex immunological reasons why HIV is difficult to make a vaccine to. But one of the major ones is that we were talking about, this is an unusual infection that establishes a latent state inside the cell. And so your immune response that's never seen HIV or hasn't seen it since you got your last shot, which is maybe a long time ago, has to get and knock out every virus that can infect the cell at the single exposure. It probably can't miss the first time, because this latent state can be established and then persist.
How common is this latent state capability?
Latent state is a part of the life cycle of the virus and happens, so the frequency of latent infection that I showed you with those dots is about 1 latent infection per every million resting CD4 cells.
But I mean in other viruses.
Oh.
If you're not talking about HIV, if you're talking
Okay.
about something else,
There's all
polio
sorts
or
of
whatever.
There's different kinds of latency. Herpes viruses as a family have a latent state, but molecularly, genetically, they're completely different from HIV. They have a latent state in that they go to sleep inside the cell to a large extent, but there's some low-level virus activity inside the cell. But because herpes viruses have lived in the human species for millions of years, we've adapted much better to them. There are viruses like hepatitis C virus that has no latent state. It has to be growing all the time, and that's why the new potent drugs that block HIV replication can cure people in 12 weeks. Hepatitis B is a different virus that has its own genome that lives inside a cell, and hepatitis B can persist for the life of a person. So it goes very specific from virus to virus.
Hi. I have a two-part question. The 3-drug ART protocol that's given to people, I know it's given to people who may be clinically exposed to HIV for a certain period of time. Is that the same protocol that's given to people who are HIV positive? I think it's called PrEP. That's the first part of my question. Are they the same drugs?
There are more than, I think, 36 HIV medications now, and they can be used in all sorts of different combinations. There's a couple of combinations that are now tested and, come in a single pill. So those several single pill triple regimens are very widely used. For PrEP, there's fewer of them that have been really studied for PrEP, which is pre-exposure prophylaxis, preventive therapy. Or post-exposure prophylaxis if you've been exposed, but the medicines that are used are the same as in general care.
Well, the second part of my question is, for somebody who may have been clinically exposed, takes the antiretrovirals and then is tested for maybe 6 months, and then is that person declared free, or could it be like the Mississippi baby and turn up 26 months later?
You can never say never. We recommend testing at 3 months, 6 months, and a year. It's incredibly rare for somebody that was negative at 3 months to be infected later.
Okay.
pretty much, it's just to be safe, but pretty much not.
Okay, thank you.
Over here, David.
Hi.
Hey.
You said earlier that in certain types of chimps, certain types of HIV don't actually have an effect. Could our knowledge of the genetic differences between humans and chimps further research in curing HIV?
Yes, maybe, and a lot of people have been working on that. So there are theories, actually they're sort of counterintuitive theories to me. Some people think that the African green monkey virus doesn't bother the African green monkey very much because the immune response that the monkey makes is almost absent. So it's as if the monkey is just ignoring the fact that the virus is there. You can easily measure the virus in the monkey. It's circulating around, but the monkey's immune system doesn't crash. It just seems to tolerate the presence of the virus. So why that actually is not really understood, but people are looking at it, and there actually had been some ideas to try to turn people into monkeys by teaching their immune systems not to respond to the virus. So far, those haven't worked out, but they're still being worked on. No, I mean, HIV is a viral infection.
Okay. Hi. This may be a difficult question, but I wanted to ask, are you aware of any breakthroughs in...
Hopefully difficult for you.
Yeah. Are you aware of any breakthroughs in treating other types of viral diseases that have occurred due to research in HIV viruses?
Well, I mean, that's an opinion question. I would say that the way we learn to test and develop antiretroviral therapy, HIV drugs, led directly to the really rapid development of hepatitis C drugs once there were a few drugs that were starting to work and were understood. Because hepatitis C therapy turned out to have to be combination therapy in the same way that HIV therapy was. And so the whole clinical testing paradigm and getting things approved by the FDA and all those sorts of pathways were really pioneered with HIV therapy. So that's at least one example.
Thank you.
I had a question. Is it unique to have an HIV Cure center locally? Are there a lot of them around the country?
I think we're the first one to call ourselves that. I can't think of another one.
That's great.
We'll have to wrap up in a couple minutes here. We'll take another question. I'll ask you a couple wrap-up questions.
One thing was, when I was in state, I'm an alumni of state, there was a guy that did it. We were in geography class together, and he did a paper on the green monkey, on the HIV hives, and that green monkey had it in his brain present, but it never caused any real problem. But what happened, that his brain was often eaten by the natives as a delicacy, and that in the process, somehow it jumped from the monkey into humans, and then it just went on to spread. And, that'd be worthy of trying, to do some research on that, and, because he had done a lot of research on that.
So that's actually exactly correct. And there's some beautiful papers. One of them's in Science magazine from about 5 or 6 years ago, showing by genetic sequencing, by figuring out how related different viruses are, that there were actually 7 times that a simian virus entered the human population. All these 7 events probably happened in sub-Saharan Africa in perhaps the 30s and 40s. That has now led to 7 different clades, general families of HIV spread across the world. So this is not really an HIV-specific thing. There are things out in the environment, and then when human society comes into contact with these things, pathogens that are hidden in a place in the wood can come out and spread. And we saw that with Ebola recently, and we've been worrying about that with flu, which may spread from pigs and chickens and other farm animals to people. So this is just essentially an effect of life that as human society is sort of everywhere all the time and we're hopping into planes and shipping things everywhere, we're essentially mixing the big pot, and we should be ready for these things to happen again.
Given all those things you just said, and so the recent scares with Ebola, and right now, I think it's lesser known how bad a situation we're having with bird flus that are killing poultry, chickens and turkeys right now. I think tens of millions are being destroyed.
Well, so this is an ongoing thing that our government does behind the scene to protect us that we don't really give them credit for. There are people all around the world monitoring the kinds of flus in different animal populations and trying to design the flu vaccine for us for next fall. So it's the right kind, because we can predict that flu spreads across the world in a yearly way, and we need to know what kind of flu it is ahead of time so we have all the vaccines made. So there's a very big operation to do that and to be able to make a lot of the right kind of the vaccine fast enough. You remember some years the vaccine doesn't work so well, or maybe there's not enough, or we guessed a little wrong. So it's a very big effort.
Do you think it's inevitable? I mean, there have been flus and plague that have killed millions of people, but it's been a while. It's been 100 years since we've seen something quite that devastating. Do you think it's sort of inevitable that this will happen again with all these emerging ones?
I'm sure that emerging infections is all the time thing, and I'm sure that's inevitable. I think we have pretty good tools. I hope that our governments are smart enough to make the investments that aren't very popular and don't really seem to be, giving you bang for the buck that year, because they're preventive investments for the long term. The Ebola is an example. So Ebola historically will go into whatever animal reservoir. We don't really know where Ebola lives for years, and then it'll pop up, cause a bunch of problems, and go away again. So now this year, the past year, we were very scared. We were trying to make all sorts of drugs and vaccines, and I hope we don't lose interest and get ready for the next time.
Maybe there's a good opportunity for maybe something like a public service announcement. We see a lot of people today are resistant to vaccines in general. And in California, in the enlightened state of California, it's actually kind of problematic. Even in the big cities, in urban environments where you wouldn't expect it. So what can you say about the fears that people have about getting vaccinated versus not getting vaccinated for the flu, for instance?
So you're setting me up. So I just think that this is where we need science education, and people need to understand what's a rational fear and what's not, and what's evidence-based practice and what's not. And, sure, people can have their opinions, but an opinion is not a fact-based thing. So let's try to decide our medical care and our public policy based on proven scientific facts. End of story.
Excellent. So we've covered it. Yeah, thank you very much. Dave, I mean, maybe that's a great ending note, but I was going to ask you, we've covered quite a lot of material, and what would you like us to leave here knowing, in addition to that?
Well, let's go back to HIV, because that's what I do. So I think HIV is a great example of mobilization of people and societies and governments over time that's made a big difference, but there's still a long way to go. It's a model for response to a lot of other similar problems. And we have made a lot of, on the slide, we've made a lot of advances. We still have a long way to go, and it will require dedication and investment. Just because it's not a crisis doesn't mean we shouldn't keep working on it.
Excellent. So thank you very much. How about a nice round of applause for David Margolis from the UNC HIV Cure Center. And thank you. A lot of great questions and comments and contributions tonight. We really appreciate it. So it's only about 8 o'clock. This building's open till 9, so you don't have to go home yet. But thank you. And next week is science trivia, and then we have some great, what's out there? It's out there now? Next week, weather and forecast. Okay, so in addition to science trivia, we'll have some cool stuff out on the floor about weather and forecasting. So thank you very much, and we'll see you next time. Good night.